Okay, good afternoon, everyone. Let's start today's class. Today is about Chapter 6, Immunological Diagnostic Technology. Okay, here is the outline for today's class. I'm going to introduce today's topic through two different major parts. The first one, I would like to introduce antigen and antibodies. That's because the basic working principle for the immunological diagnostic technology are mostly based on the interaction between antigen and antibodies. So today, first, I would like to review some basic knowledge about immunology, talking about their antigen and antibodies, how they interact with each other. And then after reviewing the basic knowledge about antigen and antibodies, I would like to talk about those immunological diagnostic technology based on this antigens and antibodies. So this is the outlines for today's class. First, let's review the knowledge about antigen and antibodies. I think that have been learned in the class of immunology. Here, I just picked some of the major concepts to help you to record those basic knowledge about antigen and antibodies. So first, antibodies can recognize and bind to their specific antigens. So here, one keywords you need to remember, that's specific antigens. So which means that the binding between antibodies and antigens are specific to each other. The antibodies cannot bind to everything. They can only bind to their specific antigens. So the binding between antigens and antibodies can be seen in vitro, which means that their interaction between antigen and antibodies after they bind to each other, then they can change for their physical condition in vitro. Sometimes their interaction can be visualized with or without the use of detecting instruments. That's the working principle for immunological diagnostic technology. That's because the binding of antibody and antigens can change the physical condition. Sometimes they get aggregation, sometimes they will get precipitate, and then we can see the change. then we can tell there is antigen or antibodies inside the system. So that's the working principle for the immunological diagnostic technology. Thus, the antigens and antibodies in their samples can be analyzed using this immunological diagnostic technology. Here shows two photos. The first one is the immunofluorescence technique. Here, as you can see, many cells inside the figure. And then we use the antibodies to stain some of the markers. If those markers are present on those cells, and then you can see the color change for the cells. So that's one example for immunological diagnostic technology. And another one is also very commonly used in the lab. That's Western blotting. This is a technique to separate proteins first and then incubate the proteins with their specific antibodies. And then you can see different bands on the membrane. So they are seen in vitro with some detecting kits. Then we can tell what proteins present in the samples. So this is two examples for the immunological diagnostic technology. And we will introduce them later. And first, let's review the concept of antigens. What are antigens? Antigen, which is the substance that is capable of stimulating an immune response, specifically activating the lymphocytes, which are the body's infection-fighting white blood cells. So based on this concept, you can learn some key points about antigen. First, the antigen is one kind of substance. That substance can stimulate the immune response in our body, especially activate the lymphocytes, for example, the B cells, the T cells. So those activated lymphocytes can react to the antigen so that protect us against to the pathogens. So as you can see in this figure, in this slide, as you can see here, there's some bacterial antigens, which means that those antigens are from the bacteria, some microorganisms, not from our cells. So this pathogen bacteria, they can release something we don't have in our body, which we call the bacterial antigen. And not only the bacteria, but also the virus can produce some of the non-self antigens inside our body. And these antigens then can activate the B cells. Then the B cells can produce antibodies, which here use the yellow dots represent the antibodies. And those antibodies can bind to the pathogen antigens, and then they can activate later the other lymphocytes to kill the bacteria or virus, so that can maintain the healthy condition of the host. So in general, there's two types of antigens recognized. One is called Fluorine Antigens, which is also known as the heteroantigens. Another one is the autoantigens, which is also called the self-antigens. Okay, we will introduce them one by one. First, the fluorine antigens. So fluorine antigens originate from outside the body. For example, they include parts of or substance produced by the viruses or the microorganisms, as we mentioned in the last slide, from the bacteria or from the protozoa, as well as some substance in snake venom, and certain proteins in foods. For example, some people may allergic to the milk, because the milk have some kind of proteins or glycoproteins which cannot digest by the people. So those proteins in food also can be fluorine antigens for them. And components of serum or red blood cells from other individuals. For example, if people have different blood types, and then can induce immunological response. So here for the fluorine antigens means those antigens produce or from outside the body, the non-self antigens in their host. So those are fluorine antigens. Mostly they are the substance from different organisms or special proteins in food or in the serum. And on the other hand, there are autoantigens. What are autoantigens? Autoantigens, on the other hand, originate within the body, which means that those antigens are produced inside our body, not from the invade of the microorganisms. They are produced or originate within our body. Normally, our body is able to distinguish self from non-self things. But in persons with the autoimmune diseases, normal bodily substrates promote an immune response, leading to generation of autoantibodies. So that means actually the healthy humans, they can tell the things from outside and inside. Usually, all the substance or all the proteins produced inside our body, our immune system should be silenced to them. They can tell those are own stuff. We don't need to response to those own stuff, right? However, if they have some autoimmune disorders, sometimes they will consider the self protein or self other particles as the non-self. They misunderstand or misjudge that they think they are not from our cells, actually, but it's their own proteins inside our body. However, the immune system make a mistake. They think they're non-self. So based on that, those normal bodily substance promote an aberrant immune response, then leading to the generation of antibodies against these autoantigens. So that's an abnormal condition and cause autoimmune disorder or autoimmune diseases. Anyway, in summary, an antigen, the concept of antigen means those substances induce an immune response and stimulate the lymphocytes to produce antibody or to attack the antigen directly. Then those substances we called immunogen, that including the fluorine antigen and autoantigens. So that's the concept about antigen. Here you need to know what are antigens and what's the major types of antigens and what they will do. They can promote immune response inside our body to produce antibodies. So next, why the antigen can promote or activate our immune system? That's because they have one specific structure, which we call antigenic determinants or epitope. That's the same, different name, but the same concept. The antigenic determinant means the part of the antigen that is the target of antibody binding. Then we call that part of antigen antigenic determinant. As you can see the figure here, here this purple structure is a bacterial cell. So as we know, the bacterial cell is not the self substance inside our body. It's from the outside, right? So for our immune system, they think this bacterial cells is the fluorine antigen. So for this fluorine antigen, there's a different shape of antigenic determinants inside and on the surface of the bacterial cell, right? Here, the red one is a triangle, and then a square, and also a half-rounded shape here. All these three different shaped structure we called antigenic determinants. What's the function for the antigenic determinants? They are the structure for the antibody binding. As you can see here, for example, this red triangle can bind to antibody A. This antibody A is produced for the binding of this antigenic determinant, and they have a specific binding. On the other hand, antibody B, as you can see here, recognize another antigenic determinant so that they can respond to the same bacterial cell. So all of this part on the antigen we called antigenic determinant. There may be one or more antigenic determinants per antigen molecules. So as you can see here, there's three different kinds of antigenic determinants on this bacterial cell. So sometimes there may be more than one antigenic determinants per antigen molecules on their antigens. And also the antigenic, when we study further on the antigenic determinants, we find that the antigenic determinants of a number of biologically important substances consist of carbohydrates, which means that those epitopes or antigenic determinants contain the substance of carbohydrates, which can activate our immune system. So this is how antigen be recognized by antibodies and why they can activate our immune system. Okay, next, antibodies. That's another concept. Antibody, which is also called immunoglobulin, it's a protective protein produced by the immune system in response to the presence of foreign substance, which means that this antibody is produced by our immune system in response to antigen. So antibodies can recognize and latch onto antigens in order to remove them from the body. So as you can see, this is a 3D structure figure to represent the binding of antigen and antibodies. As you can see here, the yellow-red one, this structure is a structure of antibody. And the blue one represents the antigen. As you can see, the antigen can bind to the antibodies and form the antigen-antibody complex. So then let's look further into the structure of the antibody or immunoglobulins. So these immunoglobulins are heterodimeric proteins consisting of two heavy chains and two light chains. As you can see in this figure, the blue-green chain here is the heavy chain for antibodies, and the pink one, the pink chain, is the light chain for the antibody. And so these two heavy chain and light chain link together here by the disulfide bonds here. That's SS means the disulfide bonds. And also these two heavy chain are linked together also by these disulfide bonds. And as you can see, for the heavy chain, there are some of the carbohydrates located in this heavy chain. And here on top of the antibodies, there's a domain or the area for the binding of the antigen. So this green one represents the antigen. So this top of the antibodies represent the antigen binding to antibodies. So this is the very symbolic figure for the structure of antibody and how they interact with the antigen. So as you can see, this structure of antibodies, so you can see their antibody looks like a letter Y, right? This looks like a letter Y. It is the basic building block for both chains. They bind together by the disulfide bonds or disulfide bridge. So to form the letter Y is the basic structure for antibody. And based on the different structure of this heavy chain, the antibodies can be divided into different isotypes, including IgM, IgG, IgA, IgE, and IgD. So here is the structure for the isotypes of antibodies. The first one is IgG. As you can see, the structure of IgG, the heavy chain for that is the gamma chain. And for the IgM, has the mu chain, and the IgA have the alpha chain. So the IgD have the delta chain, and the IgE have the epsilon chain. So these different immunoglobulins have different heavy chain structure, and also they have different oligomers. For example, the IgG, IgD, and IgE look similar, except for the structure in the constant region. But for IgM and IgA, they have different dimers, right? The IgA has two subunit, and then this is called a dimer, and this one have 5. IgM have 5 units for the structure. And for the L chain, usually have different subtype, including the kappa and llama. And this figure shows another spatial structure of the antibodies. As you can see, the top one, you can see we use the letter V for representative. V means variable region. As we know that variable means it can be changed a little bit. So this variable region have a little bit flexible for the binding of antigen. So they represents for the antigen binding region here, including the variable region light chain and variable region hairy chain. So both the light chain and hairy chain on top of it is the variable region for antigen binding. They can have their spatial flexible for the binding of antigen. On the other hand, the rest of their antibodies is called the constant region. So we use the letter C to represent for the constant region. So this constant region usually related to the biological activity of each of the isotype of antibody. So this is the isotype and also the structure of the antibodies. And here, this is a 3D figure for natural antibodies. As you can see, the heavy chain here use the red model to represent, and the light chain use the yellow to represent. So this is the 3D spatial structure for the antibodies. Okay, next, further, the scientists studied each of the amino acids for the immunoglobulins, and they learn each of the part they have different of their function. As you can see, there is 2 different antibodies here. The first A antibody means the secreted IgG, which means that this immunoglobulin, they are free in their circulation system. They can be secreted after they are produced, and then they can migrate to anywhere inside the vessel. So this is the secreted IgG. And another is the membrane IgM. As you can see that this IgM is located in the plasma membrane of the B cells, which means that they are immortalized. They are fixed on the plasma membrane of the B cells. So these 2 different types of IgG, why they have this different biological function? That's because they have different constant region, which is related to their biological activity, as I mentioned in the last slide. As you can see, the constant region of these 2 different immunoglobulin looks different, right? So there's 2 different domains, there's 3 different domains. So this structure is different. So they have different biological activities. And also, as I mentioned, on top of the light chain and the hairy chain, this is the antigen binding site. And this binding site plus the constant region 1 together, they call the FAB region. What's FAB means? FAB means fragment of antigen binding. So it helps for the antigen binding. So this FAB means this part, including the VH, which means the variant region of the hairy light, and the constant region CH, heavy light 1. So this part we call the FAB region. And the rest of the structure is the FC region. FC means the fragment crystallizable, which means that this part of the immunoglobulins can be crystallized. So we can see the crystallized protein structure for this FC region, usually including the constant region 2 and 3, sometimes including 4. So this is the FAB region and FC region. Between these 2 regions, linked by this hinge. So the function for the FAB is for antigen binding, and for FC, what's the function for that? There's quite a lot of biological function related to this FC region, including the activation of complement system, and also binding to the Fc receptor of cells, and they can passage through the precipitate and mucosa. So they have their own biological activities based on different structure of Fc region. So this is the structure of secreted and membrane IgGs. And here, if we see the top of the FAB region, you can see they have a spatial structure which can fit only perfectly to their specific antigen. So that's the reason why antigen and antibodies interaction have the characteristic of specificity. That's because they fit to each other only. Okay. See further structure of the IgG with more details. And then you can see the variable region can be further divided into different regions. As you can see, some of the parts. The first one is HVR, which represents for the hypervariable region. So based on this name, you know that they have a very high frequency of variable change for binding of the antigen. So it's called hyper variable region. And then next is the CDR. What CDR means? CDR means complementarity determined region, which is fixed to the antigen, fixed to the antigen epitopes. So this is fixed to the antigen to each other. For example, here shows the top of the antibody and the antigen. They have the exactly match for the spatial structure. As you can see here, which lights we holding the fruit with the hands. There's no other spatial for other antigens for the input. So the antigen fix the pocket of the antibodies completely so that they can bind to each other completely. That's due to the complementarity determined region in the antibodies. And this complementary determining regions is maintained by the framework region here, which is labeled in green. So you can think this framework region exhibit their function as they can maintain the spatial structure of the variant region so that the complementarity determined region can fix to the epitopes on their antigens. So this is the structure of immunoglobulins, why they can bind and why they only can bind to their specific antigen. That's because the antigen fix the pocket of their variant region on their antibodies. So here more spatial structure for the binding of antigen and antibodies. And then next question would be, now we know that there are antigens, they come from outside our body or inside our body. And the antibodies, they have the antigenic determinants, which is also called the epitopes. They can fix the variant regions of the CDR inside the antibodies so that they can fix to each other. But how the antibodies work here shows the how they works. But first, when an alien substance enters the body, so for example, the bacteria or the viruses or the other proteins, for example, from the snake venom or some proteins inside the food, all this alien substance enters inside the host, the immune system is able to recognize it. The immune system will get an alert. They know, oh, that's something from the outside, not from our host. We need to respond to it. So the immune system is able to recognize this alien substance as the fluorine, because the molecules on the surface of the antigen differ from those found in the body. So they can tell that's the non-self substance from the outside. Then, in order to eliminate the invader, the immune system calls on a number of mechanisms. They will activate a lot of mechanisms to respond to this alien substance. One of the important response is antibody production. Let's see how it produces antibody. So antibodies are produced by the special white blood cells, which is called B lymphocytes. Also, we can call it B cells for short. As you can see here, first there's some alien substance we called antigens enter inside our body. And then the BCR mediate activation. The BCR means the B cell receptor on the B cells. So this is a premature B cells actually. And after the B cell receptor recognizes antigen, and then it turns into the plasma cells, which is the mature B cells. Then this mature B cells can produce antibodies. You can see this Y letter here. This represents their antibodies. Then to mediate different activities, including, which is good for antibody-mediated protection. They can eliminate the invaders, kill their, for example, neutralize their toxins, kill the bacteria, help to kill the bacteria or the viruses. But if the plasma cells produce too many antibodies, more than is needed, and then it will also mediate the antibody-mediated inflammation, and then it will cause damage inside our body. So how to produce enough, just enough antibody, keep the balance, and can maintain their good side of the antibodies. Otherwise, overreacted can cause inflammatory injury in our body. And also those alien antigens can act and can be phagocytized by the antigen presenting cells. And then this antigen presenting cells can present those antigens to the T cells, and then the T cells can further modulating the switch of the B cells to promote more B cells activation and produce more antibodies. And these B cells can also react to the T cell activation. So the B cell T cell can also influence or affect each other in the response to the antigen. So this is how antibody produce. Then after antibodies produce, what to do next? As antibodies produced by the plasma cells, and then the antibodies enter into the vessels, they circulating in our blood system, right? Then they attack and neutralize antigens that are identical to the one that triggered the immune response. So as these antibodies can recognize those antigens that that triggers their immune system. And for example, here, our host get infected by the bacteria, and then the bacteria can produce a lot of toxic substance, which we call toxins. Here in this photo, the toxins use the red dots to represent the toxins. And if the toxins bind to the receptors on the cells, and then these cells will be dead. For example, they will cause apoptosis and do the cell damage, and then the cell will die. However, after our immune system produce the antibodies to recognize this toxin as a foreign antigen, and as you can see here, they will perform one kind of effect we called neutralization. As you can see, the antigens can bind to the antibodies and then protect the cells. The toxins cannot bind to the cell receptors on the cell anymore, and they are removed by the antibodies. And then those antigen-antibody complex can be digested by the macrophage and then later get licensed inside the macrophage. Then they are removed from our body. So this kind first protection effect of the antibodies we called neutralization. They can neutralize those toxins from the bacteria. And also the second type, this time, not only the toxin, but the whole bacteria are circulating in the extracellular space. As you can see, this represents the bacteria outside the cells. And then this is a macrophage, which one kind of immune cells. And now this time, this antibody can function through the opsonization. What's opsonization means? As you can see here, because this bacteria can produce the antigens on the surface of the bacteria, right? And then those antibodies can recognize the antigen and then bind to the antigen. So now the bacteria is covered by these antibodies, and then this antigen can promote or mediate the phagocytosis in the macrophage, and then the macrophage can digest the whole bacteria and can then trigger the lysis or bursting of the invading microbes and then remove them, protect the cells. So this is another function. And the last one, also the antibodies, what they can do. Here also, this is the bacteria in plasma. So this is a bacteria in the circulating plasma. And now the antibodies can also bind to this bacteria because the bacteria can express the antigens on the membrane, and then this antigen cover the bacteria again, and then they can activate the complement system and then induce the lysis and injections in the macrophage as well. So this is the specific antibody response by the antigens and then protect from infection. So this is how antibodies are produced and how they work. So far we have learned antigen antibody structure, the basic concepts for them, and they can get reaction, they can get interaction with each other. And for the antigen antibody reaction, there's some characteristic for them. The first one is specificity, which means that their antibodies can only bind to their specific antigens, which express the special epitopes. And their next characteristic will be reversibility. So the binding of antibodies and antigens is reversible. So what depends the binding and the dissolve of the antibodies and antigen, that based on their binding affinity, how strong the antibody and antigens bind to each other, that's the affinity. And also the environment condition can affect the binding of antigen and antibodies, including the electrocytes in the buffer, the pH value, the temperature, or affect the binding affinity of all the environment to change the binding of antibodies and antigens. So the binding of them between them are reversible. And the third characteristic will be proportionality. What does that mean? That means only suitable concentration of antibody can bind to the antigen and then cause the Wenzhou stage for the binding. What does that mean? As you can see in this figure, that represents three different conditions for the antigen and antibody binding. So the first one is that antibody exacts the antigen, which means that more antibody compared to the antigen, so too many antibody left. So as we know, the immunoglobulin, the antibodies, actually they are proteins, right? So we have learned the characteristic of proteins in biochemistry class. We know that the proteins can repel to each other because they have the water cover, and also they have their charge on their structure. So when too many antibodies there, they can repel to each other, prevent aggregation, right? So this time it's a soluble condition for antibody and antigen binding. On the other hand, if less antibody in the system, a lot of antigen inside the system, what will happen? And still, because the antigens are soluble, so in this system, still no visible precipitant can be seen in the system. So only we have a suitable ratio between the antibody and antigen, and then they can aggregate to each other together and then precipitate in the buffer, and then we can see this change in vitro. So this theory we call the lattice theory. So based on this theory, we know that when we do the immunological diagnosis and analysis, not always putting more antibody can give a good result. Only the suitable concentration of antibody can give you the best outcome. So this is the proportionality of the antigen and antibody reactions. And last, this antigen-antibody interaction has two stages. The first one is the specific binding stage, which means that the antibody can recognize and bind to the specific antigen. So this is the first stage, the specific binding stage. And then the second stage is the visual stage, which means that they can aggregate together, and then we can see the change in the buffer. That's the visual stage. That's two stages for the antigen antibody reactions. So these characteristics are very important. That can help us to design the techniques for the lab or for the clinical analysis. So here you need to understand. When you understand these characteristics better, and then you can understand the techniques, why they decide those steps, why they do so, and then you can understand the whole experiment better. Okay, we have finished reviewing the concept about antigen and antibodies. And next, let's talk about the immunological diagnostic technology. Because there's a lot of different immunological diagnostic technology, due to the time limitation in the class, I only introduced 4 types of them. The first one is the agglutination reaction. The second is the precipitation reaction. The third is the immunolabeling technique. The last is the protein microarray. I will introduce all of it one by one, okay? First, the agglutination reaction. Now, what does that mean? It means particle or soluble antigens, including the intact bacteria or some of the cells, bind with specific antibodies. The binding results in some visible agglutinates after certain time and under certain concentration of electrolyte. There's 2 types of agglutination reaction. One is direct agglutination reaction. Another one is the indirect agglutination reaction. As you can see in this figure, there's 2 different types of agglutination reaction. The blue one is the direct agglutination reaction. Here, the antigen is the particle antigen. This is the big one, the blue big one. It is the particle antigens. It could be intact bacteria, it could be cells. And these particle antigens can be recognized by their specific antibodies, which is this letter Y represents the antibodies. And these particle antigens can bind to these antibodies, and then with a suitable concentration, they can agglutinate together, and then we can see with our eyes, we can see these agglutinations reactions in their system. This is the first one, which we also call direct agglutination reaction. The next one is the indirect agglutination reaction. Here, use the red color to represent the process. Here, the antigen are soluble antigens here, which uses black rectangle represents the soluble antigens. So the soluble antigens need to bind to the vectors. Mostly, they are the vectors could be the particles or they can be other cells. Here is the red blood cells. So these soluble antigens bind to the red blood cells. Now, these red blood cells can be the vector carry its self-antigens or the soluble antigens on the surface of the cells. And then these new particle antigens can be recognized by the antibodies and then get agglutination. So this type of agglutination we called indirect agglutination reaction, because these soluble antigens need to bind to the particles or cells first to form the particle antigens and then recognized by the antibodies and get agglutination. So this need one more step. So we call indirect agglutination reaction. So for the agglutination reaction, we know the concept, we know the classification, two types. And here shows one example for the direct agglutination reaction. This is the experiment for determination of the ABO blood groups. I think if you have had your physiology class, you may have done this experiment before. This direct agglutination reaction can be performed in two different ways. One is use the slide method, another one use the tube method. The mechanism for that, for both experiments are the same. That's the agglutination, direct agglutination reactions within the rubber cells and the antibodies. But one is using the slide for the experiment. Another one use tubes. So for the slide method, we can use less sample, and the tube methods we need more blood samples for the experiment. So we can see the details about this experiment. First, this is the antigen on the rubber cells. We know that human beings have different blood groups, right? That's because they have their antigenic determinants on the rubber cells, including the antigen A, antigen B, antigen A plus B, and antigen neither A or B. So this is the type A blood group, type B, type AB, and type O, right? So this different blood type in human beings. I think you all know that. And because the human being or the patient with their antigen A on their rubber cells, and then the antigen A is considered as the self substance. So in their blood, they don't have anti-A antibody, right? But they have anti-B because these rubber cells, they don't have the antigen B. So in those people, in their plasma, we can find anti-B antibody, right? So the blood type for that is type A. Also, it can be type A. And then for the B blood type people, on their rubber cells, and then they have the antigen B. And for their plasma antibody, you can find the anti-A antibody, right? And for the antigen AB type people, and in their plasma, you cannot find any anti-A or anti-B antibody because antigen A and B are both self substance. So the body do not produce antibodies against them. And for type O blood type, and because they don't have antigen A or antigen B, so in their blood they have the antibodies against antigen A and antigen B. So when we add the rubber cells and then we add different antibodies, and then we can see the direct agglutination reactions inside the system. So this is one example for the direct agglutination reaction. So here, the patient's rubber cells can also, for the test of their Rh typing, this is a rare blood type in human being, only very rare. So for this time, we use another antibody to test for that blood type. So this is one example for direct agglutination reaction. And next is an example for indirect agglutination reaction. For this one, the example would be the pregnancy test. Here, as I mentioned before, the indirect agglutination reaction, we have the soluble antigen. And the soluble antigen need to bind to the particles first to form the particle antigens. And these particle antigens can recognize by their specific antibody, and then they get precipitation and they get agglutination, right? So this is the concept for indirect agglutination reaction. So based on that, we can do the pregnancy test with this mechanism. How it works. So for the pregnant lady, in their body, they have a high level of HCG, which represents the human carbonic gonadotropin. This one kind of, you can think it's one kind of things when you increase after the pregnancy. So because the high level of HCG in the pregnant lady, so their body will have a high level of anti-HCG antibodies. So we can design indirect augmentation reaction for the test of that. For example, the antibodies of this if the sample have the anti-HCG antibody, which we use red letter Y to represent anti-HCG antibody, and then inside the test usually make us like this, which looks like a pen. And here usually drops the samples, and then these antibodies can run through this whole path, passing first the test line and then the control line and then to the end. So this is one kind of the NC membrane for the water or for the fluid to run through. And then if the lady is in pregnancy, because the high level of HCG antibody here, which represents this red Y, and inside the tube, there is another antibody which can bind to the anti-HCG antibody, which here we use the purple Y to represent the secondary antibody. So each of the sample have the anti-HCG antibody. This red antibody can bind to the secondary antibody. Then they can run together as a complex. First, going to the test line. So what located in the test line? As you can see, there is a yellow particle here. This is the antigen, which is the HCG, right? So this antigen can bind to their anti-HCG antibody. So this red Y can bind to this antigen. And then in the test line, it will show positive line here. And later, they run through this control line. So here, control line, there are a third antibody located here, which can recognize the secondary antibody antibody, which means this green antibody can recognize this purple antibody. That's for quantity control. So if the test line, the control line run, there's no lines for the control line, means that this purple antibody is not working. So the whole system is not working. At the very beginning, there's no purple antibody here. So this is an invade result for that. So this C line means the control line to confirm the effective of the whole system. So this control line should be always positive. Otherwise, this is an invalid test. On the other hand, if the lady is not pregnant, so inside their urine, there's no anti or very low level of anti-HCG antibody here. As you can see, you cannot find a lot of the red right letter here. And then because the flu still go because you drop the sample here, then the secondary antibody will run by themselves. And then for the test line, there's no binding between the HCG to their specific antibody. So the test line should be negative. However, the secondary antibody, when they run through, because the whole system is working, so the control line is positive. So this time, this result means not pregnant. So this is how to do the pregnancy test using this indirect agglutination reaction. So this is the example for the indirect agglutination reaction. I hope you can understand the whole system. And the next reaction is the precipitation reaction. So this one is a little bit different. Here in the precipitation reaction, the antigens are soluble. So they could be the serum protein. They can be the lysate from the cells or the lysate from the tissue. And then you harvest those soluble antigens. And these soluble antigens can bind its specific antibody, and then after the binding, they are visible, precipitate in the buffer. For example, the first one is called a ring precipitation. So for this test, first we prepare the buffer with the antibody, and then we collect the sample from the cell lysates or from the tissue lysates or from the human serum. And then if these samples contain the antigen, which can be recognized this antibody, and then on the contact surface, after the binding, they will get precipitate, and then you can see a little bit blurry in this contact surface, and then you know that, oh, in the sample there's the tested antigen inside the sample. This is the ring precipitation. And for another method, we can do the flocculent precipitation. This time we mix these two samples, the tested samples and the antibody samples, we mix them totally. And then in the system, there's randomly running antigens and randomly running antibodies. So after the interaction for a while of time, and then we do the centrifuge. If they have the binding, and then you can see the precipitate at the bottom of the tube. So this is the flocculent precipitation. So there's two different ways to do the precipitation reaction. The mechanism should be the same, but for the ring precipitation, only can see the precipitate in the contact surface. And for the second type, you can see at the bottom. So that's the only differences in these two methods. Also, this precipitation reaction take place not only in solution system, but also they can be performed in the semi-solid medium, such as the agar gels. Let's see them, how they work. This experiment we call the single immunodiffusion. So how to perform this experiment? First, we need to prepare the agar plate, which is when they are hot, they are liquid, and after the frozen, after the temperature reduced, they can be solid medium. So this is called the agar plate. So this agar plate, when they are when they are in the form of liquid, we can add the antibody inside the gel. So after they are frozen, we got the antibody containing gel, which look like the jelly. Which is a little bit soft, but however, it's solid. And then this antibody containing gel, we make holes in the gels, and then we add the samples into the holes. If the samples contain the antigens, which can be recognized by the antibody, and then they will have the precipitate around the hole, right? If more antigen there, and the ring would be bigger. So when we measure the ring diameter, and then we can calculate the concentration of antigen inside the sample. So this is the single immunodiffusion. This experiment is easy. Just prepare the gel containing the antibody, and then make a hole in the gel, and then add the samples. If they can react to each other, you can see the ring around the hole, and then by calculating the diameters of the ring, and then you can calculate back for the antigen concentration. So this is called single immunodiffusion. And also, if we cut or we make two holes in the agar gel, and then this we can, so make three holes on the gel, and then we can do the double immunodiffusion. So what's this double immunodiffusion for? And as we can see, the first condition, there's two holes for antibody, two holes for antigen, one hole for antibody. So the antigen holes, we use the blue holes to represent for that, and the yellow hole for the antigen. The yellow hole for the antibody. So in the first hole, it contains antigen 1 and antigen 3. For the second hole contains antigen 1. And then for the antibody hole, we add antibody 1, which can react to the antigen 1. So what will happen? Because these two holes all have the antigen 1, right? So they can respond to antibody 1. So here, as you can see, they can precipitate for both holes. So you can see two lines here. Because these two antigen holes contain the same antigen, which can respond to the antibody. So this is the first condition. These two holes include the same antigen. So for the second type, now this antigen holes, the first one I put in the antigen 1. The second hole I put the antigen 2 and antigen 4. And for the antibody hole, I put the antibody against antigen 1, 2, and 4. And then you can see the results look like this one. The antigen 1 will react to antibody 1 to make the first line to the left. And then the antigen 2 and 4 can react to their antibodies 2 and 4. Because antigen 1, 2, and 4 are completely different antigens, so the reaction should be completely different. So as you can see here, the three lines, the shape of the three lines are completely different. So based on this experiment, we can tell the antigens in these two holes are completely different. And for the condition 3, now the two antigen holes, the first one I put in antigen 1, the second hole I antigen half 1, half 2. And then this antibody hole, I put the antibody against 1 and 2. And now, as you can see, the antigen 1 can respond to antibody 1 to form the first line. And then the second hope, because they contain at least half of the antigen, react to their antibodies. And then this line looks a little different compared to the condition 1, right? So this kind shape of lines represent that in these two antigen hosts, they have pathway the same antigen. So some of them are the same, but not completely the same. So this condition 3. So by using this double immunodiffusion, when we get the results for different lines, we can do the both the quantitative and quantitative analysis about the antigen. So this is the double immunodiffusion reactions. Okay, we have finished the agglutination, the precipitation reaction. And next, let's talk about the immunolabeling technique, which is commonly used in almost all the labs. So what are immunolabeling? So immunolabeling is a laboratorial technique used to identify antigens of interest in a sample. This can be useful for diagnostic as well as research. So it can be used not only in the labs, but also in the hospitals. So what kind of things can be labeled to the antibodies? It include the enzyme. We can use the luciferin, or we can use the radionucleoside, and also we can use the chemiluminescence substance for the immunoneighboring. And all of them can play an important role in clinical trials, the epidemiological study, and evaluation of specific orders. So, sorry, why there's so many noises? Okay, so properly labeled samples can be examined under a microscope or other instrument to collect or record information that may be relevant to a scientist's work or medical diagnosis. So let's see more details about the immunolabeling. So the first one, it is almost the most commonly used techniques in all the labs and the hospitals, which is called the enzyme immunoassay. Another name for that is the enzyme-linked immunosupplement assay. We usually call them ELISA, E-L-I-S-A, ELISA, in which an enzyme bound to an antigen or antibody functions as a label. Someone work on the microphone. Could you please silence it? There's too many noises, too many background noises.
Ma'am, can you please do that? We cannot.
I think someone turned on their microphone, so they produce a lot of background noises. Okay, I don't know who turned on the microphone. Anyone? If you are in the class, could you please turn off your microphone so that to avoid the noises? So it's not caused by my microphone, so I cannot reduce the noises. Okay, so the ELISA, there's different types of the ELISA, including the indirect ELISA, the salvage ELISA, competitive ELISA, and the biotin aridin system ELISA. So all of them are commonly used in the lab. So I will introduce them one by one. So the first one is indirect ELISA. I think this one is easy to understand. This is the well of the plate. And in this well, at the bottom, as you can see, we coded the antigen on the bottom of the well. So it's the antigen coded well. Then we wash the well to remove the loosely bound antigens in the well. And then we add the samples inside the well. So if the sample contains the antigen, which can respond to the sample, if they contain the antibodies which can recognize the antigen, then they will have the binding, right? So if the sample have the specific antibodies, they can bind to the antigen which is coated in the bottom of the well. And then we wash the sample again to remove the loosely binding or the non-specific binding. And then here we add the secondary antibody here, which is labeled in black. This secondary antibody, which is linked with the enzyme here. And then this enzyme, when we put in the substance inside the well, then the enzyme can catalyze the reaction of the substance. And then, as you can see, the color of the well will be changed, right? So at the very beginning, without the antibodies in the samples, the well looks the color blue. After we add the samples and the testing secondary antibody and the substrate, and then it turns into yellow. So when we tell the differences between the wells, the color differences in the wells, and then we can know that the samples contain the testing antibodies. So this is the indirect ELISA. Coded antigen, add sample, and then add the secondary antibody, and then they have the color change. And then we know that inside the samples, there is specific antibodies there. So this is the indirect ELISA. And next type is the salvage ELISA. What that happened? First is also the well of inside the plate. And then at the bottom of the well, this time we are not going to coded antigen, but here we coded the well with the antibody. Here we use blue Y represent the coded antibody inside the well. And then we wash the well, and then we add the sample. Then if the sample has the specific antigen, here we use the yellow bars to represent the antigens. So if the sample contains the specific antigen, and then they can bind to the antibody, right? And then we add another secondary antibody which link with the enzyme, and then we add the substance as well, and then the color of the well will be changed again. So for this salvage ELISA, we are testing the antigens in the sample. And for this indirect ELISA, we are testing the antibodies, which is labeled in blue here. We test the antibodies in their samples. So that's the differences between the indirect ELISA and the salvage ELISA. So when you choose the indirect ELISA, when you choose the salvage ELISA, that depends on the sample you have. If in the sample you want to test the antibody, then choose the indirect ELISA. On the other hand, if you want to test the antigens in the sample, then you can choose the salvage ELISA. However, these two types of ELISA have a little trick for the antibody, the choice. The first one here, when you choose the secondary antibody, as you can see, this secondary antibody reacts to the sample antibody. So when you choose the antibody, make sure that you choose the correct antibody which can respond to the testing antibody. However, as you can see for the salvage ELISA, the secondary antibody, do they react to the primary antibody? No, they don't. Instead, this secondary antibody also bind to the antigens in the samples, right? So doing different types of ELISA, it's very important to have a clear mind that what I'm going to test, antibody or antigen. That will lead to the choose of different secondary antibodies. If you choose the wrong antibodies, and then you will may have a false positive or false negative results. So that's very important. Make a clear idea what I'm going to do and what should I do. That's you need to figure out before the experiment. And next would be the competitive ELISA. So for this competitive ELISA, as you can see, first the well is labeled with the primary antibody. And for this experiment, we are testing the antigens inside the sample. However, we also add of some of the labeled antigens as well. And then if the sample have no testing antigens, all the labeled antigens will bind to the primary antibodies, right? And then you will have a very strong signal data for the ELISA. However, if you have the testing antibodies, which is not labeled, this is from sample, this you add to the system. And then the more antigens in the samples, then the lower signals you can get back from the system. That's competitive ELISA. So for this kind of ELISA, you add the same antigen. However, those added antigens you are labeled, you label those antigens, and then the testing antigen and the label antigens will competitive to bind to the primary antibodies in the well. And then by testing the signals from the label antigen, then you can calculate back how many testing antigens in the system. This is the competitive ELISA. Why we need the competitive ELISA? That's sometimes only very few antigens inside the system. So using the sandwich ELISA maybe give you a very, very low signal. So for that, to make the signals more easy to be detected, so you use the label antigens to competitive binding to the antibody, because this you can add a lot, right? So based on calculating the labeled antigens, and then you can calculate back the testing antigens in the sample. So that can help you to easier to measure the testing antigens inside the system. So this is the competitive ELISA. Again, if the testing sample have a very, very low feedback, then you can also use the biotin evidence system, which we call the BSA. As you can see, the BSA, how it works. First, the antigen labeled in the or coated in the well, and then the specific antibody can recognize the antigen for the binding. However, if the antibody are too few of them bind to the antigen, and then the signal will be very weak. However, if the antibody are labeled with the stravadine enzyme coated here, this is a coated enzyme which can be recognized by its aridine. So this biotin avidin can specifically bind to each other. So after binding of the antibody, this one antibody can bind to a lot of the stravadine enzyme, so that they can put this signal bigger, because at the very beginning, only one signal for this antigen. However, after more binding this stravadine enzyme coded system, and then you can maximum the whole signal in the well, so that you can test the antigen or antibody much easier. So this is the biotin evidence system, BSA, for ELISA. So this is the 4 different types of ELISA experiment, which are commonly used in almost all labs. In my labs, this one, sandwich ELISA, and the first one, indirect ELISA, are very commonly used. For the competitive ELISA, not that common because it's complicated for calculation, so my students don't like it. And for this belting evidence system, sometimes we use that, but this one very expensive. So if we can get the results from the previous two ELISA, we don't use this one because we don't have that much money. Okay, next technique is the immunohistochemistry technique. So this technique includes the application of histochemical and immunological methods to chemical analysis of the living cells and tissues. So for the ELISA testing, the purist technique, we only test the soluble, sometimes soluble antigens, sometimes soluble antibodies. However, sometimes our samples are from living cells and tissues. As you can see here, this is the tissue section from the hospital. For example, we want to know some kind of proteins, the expression level of the proteins inside this tissue, then we cannot do the ELISA because they are not soluble. It's a solid section from the, for example, the cancer tissue, right? So this time we can use the immunohistochemistry techniques for the detection. How to do that? First, we have the tissue or cells in the slide. And in this tissue or cells, they can express some kind of proteins, some kind of antigen. And then we use the specific antibody to recognize this antigen. As you can see here, the primary antibody, which is the red letter Y, can recognize this antigen, which is also one kind of proteins. And then they have a specific binding. And then next, we use a secondary antibody, which can bind to the primary antibody. And this secondary antibody, we have the HRP label, which is one kind of enzyme. This HRP is the horseradish peroxidase, which is one kind of enzyme. So as we know, the enzyme can characterize some of the chemical reactions, right? So this HRP, this is the enzyme, we add in the substrates here. DAB is the substrate for this horseradish peroxidase. So they can turn this purple DAB into the brown color products. So when they go into this section, we can see the negative regions, they have a normal color. However, the positive regions, those cells or those regions expressing the antigens, they will turn into brown. So this is the technique of immunohistochemistry analysis. So this technique can be used for the cell section or tissue section, and they can give the information about how many proteins or antigens are expressed in their section, and also they can tell you the location of the expression. So this technique is also very commonly used in the labs and hospital. In the hospital, especially for the cancer diagnosis, this technique is used every day almost. And next is the immunofluorescence technique. So this time, the labeling of the antibodies or antigens, we label them with the fluorescent dyes. So as you can see in this figure, they have the fluorescent emissions from the sample. So they can be used for the purpose of demonstrating the presence of a particular antigen or antibody in a tissue preparation or smear. So here, the secondary antibody labeled with the fluorescent dyes. So under the microscope, you can see this very beautiful staining color. For this example, the green color, they used elixir 488 as the staining, which labeled to the antibody anti-alpha tubulin. So those green color means the presence of the alpha tubulin. And then the red one, they use the dyes of DAPI. That's another dye for the nuclear staining. So the red dots represents the nucleus inside the cells. So this fluorescent immunofluorescent technique is similar to the previous immunohistochemistry technique. However, for this immunohistochemistry techniques, you can only see one color, the brown color. But for the immunofluorescence technique, you can stain a lot of color. In my lab, the students try to use more than 3 colors for the staining, so that give you very beautiful pictures or photos. You can see different location of the proteins or the nucleus. So that's more tell you more information compared to the immunohistochemistry staining. This one is maybe better. However, they still have their limitation. The fluorescent signals depend greatly on the quantity and concentration of the antibody, the proper handling of the specimen, and detection with the appropriate secondary antibodies. And also, this fluorescence dyes, fluorescence was gone will be gone very quickly. So if you cannot take the photo within half an hour, they will decrease the signal very quickly. So then maybe you get the false negative results. So doing this experiment give you a better picture, but they have a higher requirement of techniques compared to this previous immunohistochemistry technique. So that's the difference for this immunohistochemistry technique and immunofluorescence technique. So both of them can deal with the cell or tissue section. However, they have different advantages and disadvantages. When you do your experiments with the tissue or cell samples, you need to decide which one is better for your experiment. Usually, immunofluorescence technique is better. However, the antibodies are more expensive and you need a better technique for the experiment. Okay, next would be the radioimmunoassay, RIA. This technique of immunoassay in which radioactive chasers are introduced into the substance to be analyzed. Here, this one, you can see the photo here. The left is the antigen you want to test in their system. Here we use the blue dots represents the antigens in the sample. And then to the right is the red antigens labeled with the radio substance, which are radioreactive to the instrument. Here we use the red dots to represent for this labeled antigens. And then we have the antibody which can specifically bind to these antigens. So first, we don't add the sample, but we add all the label antigens with the antibody. And then we will have the bound between the label antigens and the antibodies. And we have a signal here. And then when we add the sample, the different concentration of the samples into the system, these sample antigens can competitively bind to the antibodies, right? So with more antigens in the samples, some of the antibodies bind to the label antigen, some bind to the sample antigens. So they have a competitive binding to the antibodies. So the binding and free signal will decrease once the antigen in the sample increase, right? So then we can write the curve here. Then we can calculate that the concentration of the unlabeled antigen will be increased once the BF ratio decrease. So they have, based on this curve, we can calculate how many antigens present in their samples. Next technique will be immunoblotting. We also call that Western blotting. This is very, very commonly used techniques, I think, in all labs. So this is a blot consisting of a sheet of nitrocellulose or nylon that contains spots of protein for identification by a suitable molecule probes and is used especially for detection of proteins. So let's see how it works. Usually, we have a protein lysates. It can from the cells, it can from the tissues, or from the biological fluid of the human beings. And then we collect the protein lysates, and then undergo the sample denaturation to denature the proteins, and then add the protein samples in the polyacrylamide gels, and then undergo the electrophoresis to separate the proteins. So this is the protein mixture. And then after the electrophoresis, they can separate it into single proteins in the gel. And then transfer the proteins from the gel to the membrane, and then incubate the membrane with their specific antibodies. And then in the membranes, we can detect the bands for specific proteins. So the results will look like this. This is a membrane. After the antibody binding to the proteins, you can see if the samples contain specific proteins, then they will have a positive bands in each of the link. So this is the Western blotting. The main mechanism for this study would be protein bind to their antibody and then visible with this technique. So the steps for the Western blot include the sample preparation. It can be the cells suspensions or the tissue suspensions. It can be breakdown with the high frequency sound. It can use the specific detergents to make holes to release the proteins, and you can use the force to make cells through small holes using high pressure, or you can sear cells between a close-fitting rotating plunger and the thick walls of a glass vessel. All these methods just want to release the proteins from the cells or tissues. And then you do the proliferation and to collect the protein sample. Then the protein sample, you need to do the quantification to measure the concentration of the protein. Because for the results, you need to compare each link of the proteins. You want to know if after the treatment, the proteins is decreased or increased. So you need to make sure that you load the same concentration of the total proteins in each well so that you can compare the density of the bands. So the protein quantification is required before the whole process. And then you get the protein sample, you treat them with the SDS, the sodium declined sulfate. So to make the proteins denature, which means that at the very beginning, the protein have their three-dimensional structure. After the denature, they will turn into the structure with only the single structure with the negative charge. And then these negative charge proteins run in the page, which is the polyacrylamide gel electrophoresis. So after this page, which we can separate the mixture proteins into single proteins based on their size. Usually small proteins run faster, large proteins run slowly, so they can be separated into the links. So from the bottom, the small protein, from the top is the large proteins. So this is the equipment for the SDS page. And next, for the transfer, as I mentioned, the gel is located in the middle, and then we have a blotting membrane attached to the gel. And from the top and the bottom, we will have 3 filter papers, and then the sponge, and then we have different electrodes. One is the positive electrode, another one is the negative. And then we can transfer the proteins from the gel to the blotting membrane. And then the next step would be in the membrane, the proteins are already there. And then we add the antibodies. And then after the after adding the substance, then we show the bands for the proteins. Then we can finish the Western blotting experiment. So using this technique, we can detect if the proteins are present in the samples, how many of them are inside the samples. So this Western blot are very commonly used in most of the labs. And next would be the luminescence immunoassay, which we also call LIA. So these are this luminescence immunoassays has been developed utilizing the luminance substance labeled antibodies to analyze potential antigens. As you can see here, the antigens is here, is the red triangle, and then this is the antibody, the primary antibody, which is labeled in blue. And then the secondary antibody labeled in yellow. A secondary antibody, which labeled with the luminescence substance. Then after the chemiluminescence substance acting in it, that will give the light emission in their system. So this chemical or substance could be chemicals, it could be biofactors, it could be the enzymes, and also it could be electrochemicals. As you can see here, the first example is the chemiluminescence substance, and it could be the biochemicals, for example, the luciferin, and it could be the electrochemicals, which can release the light emissions after the stimulation of the electrodes. And also it could be the enzymes, as we mentioned, the horseradish peroxide. So after label the antigens with this luminance substance, we can detect the lights from the reaction so that we know the binding of this substance. So the last would be the protein microarray. So this one, I think we have introduced in the previous class. This protein microarray is the protein chips. I think I introduced this in the second class. So as adding the samples inside the wells, we can perform the high throughput assays so that we can get the information, what type of proteins inside the samples, what's their function. So using this, we can perform the protein microarray. So this experiment a little bit expensive. However, it can give a lot of data based on one simple test. So still, it's very commonly used in China. So that's all for today's class. As usual, if you have any question, you can send me email to ask. Thanks again for your attention. That's all for today's class. Thank you so much. Okay, bye-bye.